Hello everybody, this is Mr. Kimmel with another screencast for Life Science. In this lesson, we are going to learn about the scientific method. To learn how the natural world works, people use what is called the scientific method. The process has several steps to it. There are 6. 1 is observe, 2 hypothesize, 3 experiment, 4 analyze, 5 conclude, and 6 communicate. Step 1 of the scientific method is to observe, or to make observations. An observation is a perception made about the natural world that causes you to ask a question. This first step also includes performing research. So notice things about the world, but also look up some information about that topic as well. All of this knowledge will help you come up with one or more questions. For example, I might observe butterflies and flowers in my garden. I observe that butterflies visit certain colors of flowers, but not others. These observations lead me to ask a question. Why do butterflies visit certain colors of flowers and not others? While researching butterflies and flowers, I come up with another question. How do butterflies eat? Please note that the best questions asked in science are why and how questions. Once you have made some observations and formed a question from them, the next step is to hypothesize. A hypothesis is a proposed explanation for the way a particular aspect of the natural world functions. More simply put, a hypothesis is an answer to the question you asked. Another way to put it is a hypothesis is a testable explanation to a situation. Let's look at the first question I come up with. Why do butterflies visit certain colors of flowers and not others? So now I want to hypothesize why this is so, or write an answer to my question, which will be my hypothesis. So my hypothesis is if butterflies are given 3 different color choices of flower, then they will prefer red because it is a bright color. It doesn't matter if my hypothesis is correct or not. That is the whole point of this method. I might be right or I might be completely wrong. That's where step 3 comes in. Experiment. Step 3 is to experiment. An experiment can be defined as a procedure carried out to test a hypothesis. While experimenting, you gather data. This data is usually numerical, but sometimes it is not. Experiment is designed to have many parts. If you're missing one or more of these next few parts, then your experiment will not be valid. First are the variables. The independent variable is the one I control. It is what you are changing or manipulating in your experiment. You can only have one independent variable per experiment. The dependent variable is what happens as a result of the independent variable. It is what happened because of what you changed. It is also what you are measuring in your experiment. Next are the groups. The experimental group is the group that I am testing with the independent variable. The control group is the group that you are not experimenting on or testing. You cannot tell if your experiment did anything if you don't have anything to compare it to. The experimental group is compared to the control group to see if there was a result. Lastly are the constants. Besides the independent variable, everything else must remain the same. These are called the constants. In our butterfly flower experiment, the independent variable is the color of the flowers available to the butterflies. This is what I am manipulating or changing. The dependent variable is the color of flower that the butterflies prefer. Recall my hypothesis. If butterflies are given 3 different color choices of flower, then they will prefer red because it is a bright color. Notice that the independent variable and dependent variable are found in the hypothesis. In my experiment, the experimental group is the red flowers. The control group is the white flowers. The constants are the type of flower, the number of flowers available to the butterflies, and the environmental components the flowers are found in, such as soil, sunlight, temperature, or anything else. Now that I've designed my experiment, I'll run the experiment and gather data. The way I would set up this experiment is to have several plants with red flowers, let's say about 10, and the same number with white flowers and blue flowers. These could be planted in the ground or in pots as long as they have the same environmental conditions. Next, I would gather data. I would keep a data table marking each time a butterfly visited each colored flower. I would do this several times over several days. The important thing about gathering data is that the more you can get, the better your results will be. Here's an example of a data table. I observed the butterflies and flowers for 7 days at the same time each day. I made tally marks under each flower color as it was visited by a butterfly, and then totaled each for that day. Gathering data is part of any experiment. These are the results of the experiment. Now that we've gathered data, we analyze the results. This is where we make sense of the data and put it into the form of a graph. First thing I'm going to do to analyze my results is to get the average number of visits for each flower color and add that information to my data table. The red flowers had an average of 15.4 visits. The blue flowers had an average of 10.7, while the white flowers had an average visit of 14.7 visits. Now I graph my data and look for trends. Here I chose a bar graph to represent the number of visits each day. Notice at the end I also include the averages. Students use the wrong type of graph to represent their data. A bar graph, like I used here, is used to compare things between different groups. A pie chart, as shown on the left, should be used to show parts of a whole. A line graph on the right should be used to show changes over time. After analyzing your results, make conclusions from them. A conclusion is the written discussion about the results of the experiment. Do your results support or contradict your hypothesis? It is important to point out that experiments don't prove whether your hypothesis is right, but they can prove that they are not right. Experiments support our ideas until new data comes up that doesn't. The other thing to add to your conclusion is to discuss things that you could do differently next time. Would you collect more data? Would you use a bigger sample size? Would you test a different variable? Look at your data and results and see what you can tell from it. In my butterfly flower experiment, I see that the average number of butterflies that visited the red flowers is 15.4, which rounds to 15. The average number of butterflies that visited white flowers is 14.7, which also rounds to 15. These numbers are so close to each other that I can conclude that butterflies do not prefer red flowers over white flowers. Even if the averages were 14 and 16, I would still say they do not show preference for one over the other. If the averages were 4 and 30, for example, then I would conclude that they do prefer one over the other. In my written conclusion, I would also discuss things I might do differently next time. Maybe I would test different colors besides red and white. Maybe I would test a different type of flower. Maybe I would collect data at a different time of the day. Anything goes. Remember that in science, we think and share our ideas with others. Final step of the scientific method is to communicate your experiment and results. Recall that science is a community and it shares information with others. Communication can be in the form of a report, a published scientific paper, giving a presentation, or making a poster or other display to share with others. Recall that the scientific method is a process. It's also not a process that flows in one direction only. Sometimes you have to back up and make a change. Sometimes you have to start completely over. The important thing, you are doing science. It's a method, it's a process, it can be tons of fun. People learn by doing things, so let's do some science and learn some cool things along the way.